What if we could stop fighting the symptoms of a disease and actually fix the problem at its source? Not with a pill, not with surgery, but by literally rewriting our own biological code. Well, today we're jumping right into the incredible world of gene therapy, a field that's doing just that, using our genes as medicine. All right, so here's how we're going to break it all down. We'll kick things off with the fundamental promise of gene therapy, then we'll get into the two main ways it's done, either inside or outside the body. After that, we'll explore the toolbox scientists use to deliver the genetic fix, see it in action with some amazing real-world examples, and finally, we'll tackle the huge challenges and the really big ethical questions that come with this kind of power. So let's start with that massive central idea. Think about it. So many diseases, everything from cystic fibrosis to certain kinds of cancer, often boil down to a tiny mistake in our DNA, a single defective gene. Gene therapy asks this revolutionary question: what if we could just go in and correct that mistake? And at its heart, this is what we're talking about. It's a total game changer for medicine. Instead of just managing symptoms, putting a Band-Aid on the problem, the whole point here is to alter a patient's genetic material to fix the root cause of an illness. It's an incredibly precise, targeted way of thinking about healing. Now, when you hear gene therapy, you might picture scientists in a lab, snipping and pasting DNA like they're editing a movie. And while that kind of thing is happening, the most common strategy is actually a bit simpler. It's more like adding a new corrected scene. You're not cutting out the old broken gene, you're just inserting a brand new working copy. And that new gene just overrides the faulty one, making the right protein and getting things back to normal. Okay, so that's the what. But what about the how? How do you actually get this new gene into a person's cells? Well, it turns out there are two main strategies for doing this. You can either work on the cells outside the body or send the fix directly inside. And this image right here lays it out perfectly. You've got two main roads. On one side, ex vivo, which is just a fancy way of saying outside the body. You literally take the cells out, work your magic, and then put them back in. Then you've got in vivo, inside the body, where the genetic package is delivered straight to the patient. Let's dig into that first method, ex vivo. The ex vivo process is this really careful step-by-step procedure. First, doctors take out some of the patient's cells, often from their bone marrow. Then, in the lab, they grow these cells and introduce them to a special, harmless virus that's been engineered to act like a delivery truck, carrying the therapeutic gene. Once that gene is safely inside the cells, they let them grow and multiply, and then you just infuse these newly superpowered cells back into the patient. So why go through all that trouble? Well, it all comes down to one word: control. When the cells are outside the body, sitting in a lab dish, scientists can check everything. They can make absolutely sure the new gene went in correctly and that the cells are healthy and working as they should before they go back into the patient. It's a massive safety check, a crucial quality control step. So whether the therapy happens inside or outside the body, you still run into the same huge problem. A cell is like a tiny fortress, right? It's got walls and defenses. So how on earth do you get your genetic package past security and into the command center, the nucleus? To solve this, scientists have developed a whole toolbox of what they call vectors. And these delivery tools basically come in two flavors. First, you've got viral vectors. This is brilliant, really. They take a virus, strip out all the nasty stuff that makes you sick, and then use its natural talent for invading cells as a delivery service. Then you have non-viral vectors, which use chemistry or even a bit of brute force to get the job done. It's a classic trade-off. The viruses are incredibly good at getting in, but the non-viral methods are often safer and less likely to trigger your immune system. And when you look at these different viral delivery vans, you see it's not a one-size-fits-all deal. Each one has its own special skills. Take retroviruses. They can stitch the new gene right into your DNA for a permanent fix, which is amazing, but they only work on cells that are actively dividing. Then you've got adenoviruses. These guys can get into almost any cell, but the fix is temporary, and your immune system can sometimes overreact. It's all about picking the right tool for the job. Now, if we switch over to the non-viral side, this is where things get really wild. Check this out. It's literally called a gene gun. And yeah, it's pretty much what it sounds like. Scientists coat these tiny little gold particles with the good DNA and then just fire them at high speed right into the cells. It's brute force for sure, but hey, sometimes it's exactly what you need. For a more subtle approach, there are these things called liposomes. Just picture a tiny bubble made of fat. The therapeutic gene gets packed safely inside. Now, since our own cell walls are also made of fat, this little bubble can just merge right into the cell and deliver its cargo. Super clever, right? It's like sneaking a message inside a care package. Okay, so we've got the what and we've got the how. We know the basic idea and we've seen the toolbox. But what does this all mean for actual people? Let's talk about where gene therapy is moving out of the lab and into the clinic, changing actual lives. And the list of conditions being targeted is just remarkable. You have diseases caused by a single faulty gene, like cystic fibrosis or SCID. You might know that one as bubble boy disease. But it goes way beyond that. Scientists are also taking aim at inherited blindness, some types of cancer, and even really tough neurodegenerative diseases like Huntington's. For that one, the goal isn't even to add a new gene, but to switch a harmful one off. And this right here is where it all comes together. This diagram for treating SCID is the perfect example. See? It's that ex vivo process we talked about. They take out the bone marrow cells, they use a retrovirus as the delivery truck to install the working gene, and then they put those corrected cells back in to rebuild the patient's entire immune system. This has been one of the biggest, most life-changing success stories for gene therapy. And let's be super clear, this isn't some science fiction fantasy for the future. This stuff is real, and it's happening now. In fact, way back in 2003, a gene therapy called gendosine was officially approved in China to treat certain cancers. That was a huge deal. It was the moment gene therapy officially graduated from being an experiment to being a real, approved medicine for patients. But for all of this incredible promise, it's not a magic bullet. Not yet, anyway. The road ahead for gene therapy is still full of bumps. Not just scientific hurdles, but some really deep, really tough ethical questions that we all need to start thinking about. This slide does a great job of showing that constant tug of war. On one side, the limitations are very real. Sometimes the effects don't last. The body's immune system can fight back against the delivery viruses, and there's always a risk of causing new problems. But then you look at the other side, and the progress is just breathtaking. Better delivery systems using nanotechnology, people actually getting their sight back in clinical trials, even curing deafness in animal models. The field is moving so incredibly fast. And then there's the question that goes way beyond the lab. These are custom-made, cutting-edge treatments, which means they are astronomically expensive. And that forces us to ask a really uncomfortable question. If we develop these miracle cures, who actually gets them? Who can afford a price tag that can be millions of dollars? And what does that mean for everyone else? And believe me, the ethical rabbit hole gets even deeper. There's a massive, massive line between what's called somatic therapy, that's fixing the cells in one person, right here and now, and germline therapy. That's where you'd be changing egg or sperm cells. If you do that, the change you make doesn't just stop with one person. It gets passed down forever through their kids and their kids' kids. Suddenly, you're not just treating a patient, you are editing the human gene pool. Which brings us to maybe the biggest, most complicated question of them all. Once we have the power to correct our genes, we have to define what's broken. What's the real difference between a disorder that needs curing and just a human difference? What does normal even mean? And maybe most importantly, who gets to decide where that line is? As this technology gets more powerful, that's a conversation that has to happen outside the lab. It's a conversation for every single one of us.